And what do you think? Should I start? Okay, I think we will start. I know that sometimes it takes a while for other people to get on. So first of all, really welcome everybody who's joined us. I know it's morning for some people, afternoon for others, and probably evening for others. So thank you very, very much for joining us. My name is Durham Wong Rieger, and I am chair of the Council for the Rare Diseases International, and also president and CEO of the Canadian Organization for Rare Disorders. And this is being brought to you by both of our organizations, and also really want to support everyone for making the effort to be here. And really thanks again to our sponsor, Pfizer, for providing the educational grant for this. So this is the third in the series of advanced therapies that we have presented. And this one is specifically on lentiviral cellular and gene therapies. And we're going to end up with a discussion around the global access to gene therapies. Next slide, Ange. So I won't go into this much. Most of you have been here and know kind of the overall objectives, really wanting to make sure that patients are beginning to become much more aware of the opportunities around cell and gene therapies, providing a bit of a background in education, but also some opportunities to hear from healthcare professionals who are providing the therapies and also patients. Next. So this one is, as you say, is really specifically focused on lentiviral cellular and gene therapies. Next. And the objectives here are, as we have in the past, really provide you with some background or education understanding of advanced therapies. Again, this time specifically on the role of lentiviral viruses, and also then you'll hear from two patients who have actually received CAR-T and Fabry's disease therapies using this technology, what it's meant to them, and maybe some things that you want to take home in terms of messages around it, and also going to discuss some of the ethical issues around getting global access. This was the long list of pre-work that we gave you, and hopefully if you didn't get a chance to go through it before, that you will in fact take the opportunity to look at it again. It will certainly, prep you a bit for what's happening today, but also reinforce what we've had before. Next slide. So this is then the presentation, as we've talked about. We're really, really delighted to have here Dr. Chris Mason with AvroBio, who's going to be speaking about the lentiviruses, and who's going to also give us some background and introduction in terms of the therapies, how they are used, and what they're used for. And then we'll have testimonials from two of the patients. So this is bio for Dr. Mason. This is the, I won't read it all, you can read it, and you can certainly have the slides and read it later, but as you can see, he comes with a huge background for us, has gone from around the world, and has landed now, I think he is in Boston, and really, we're really so very delighted that he's taken the time to get up and we're speaking with us live today. We'll be available for R&D, for Q&A afterwards. Next. Just briefly then to the patient who will be talking about their experiences in receiving cell and gene therapy. One of them is Brian Kaufman, who's going to speak to you in his capacity actually as a cancer patient. And the other one is Darren Badulka, who is the first patient with Fabry's disease to receive gene therapy. And he's going to talk to you about what his experience has been over the past 5 years. Next. So we'll do a bit of an exercise afterwards, and really we're going to focus on talking about what is it like to, what are the challenges around global access to these therapies, because I think we're all pretty amazed so far in terms of what they're able to provide. And then we'll end up with a discussion back with everybody. Finally, I think that's it. Okay, so without much further ado, we're going to turn it right over to Dr. Chris Mason, who is so kindly here with us. And I think they'll be sharing their slides directly. If you have questions during, please do put them into the chat box. But Dr. Mason's going to stay with us, and we'll have an opportunity to chat with him live at the end of it as well. So thank you so much for being here. Thanks to AfroBio for making you available. Thanks to Lori and Fernanda for helping us pull this really, really, interesting session together. So over to you, Chris.
Thanks, Doreen. That's really fantastic. And a big thank you to Cord for actually organizing this important webinar series, and the sponsors and other contributors. And I'm really delighted to be here. I've had 30 years in cell and gene therapy, and for me to be able to share some of my experience over that time with your community on cell and gene therapy. And my session today, as you quite rightly said, will focus on lentiviral gene therapy and why I believe it's important. So if we could have my first slide, please. So I must just say that the therapies that I'm going to talk about today for lysosomal storage disorders are still investigation, and they have not been approved by any of the regulatory agencies. And so what we're looking at really is clinical trial data and preclinical data. And I'm going to discuss basically lentiviral vectors in general. I'm going to discuss one particular aspect, which is important for the therapy. And also I'm going to discuss our programs, and then I'll summarize and we'll go to Q&A. All in 20 minutes. Okay, so introduction to AfroBio. Next slide, please. So we're a company that we established. I was one of the co-founders in 2015. And really what our purpose is really to free patients from a lifetime of genetic diseases, mono-inherited diseases. And we've chosen lysosomal storage disorders. And our vision from the get-go was to discover great science and then enable it to be translated into benefit for patients. And in fact, our very first science came from Toronto, from UHN in Toronto for Fabry. If we go to the next slide, please. And to date, we have 3 therapies which are going through clinical trials, so still being investigated: Fabry disease, Gaucher type 1, and cystinosis. And that's about a total of 16 patients we've treated so far. And then we've got 3 programs which are still in the preclinical stage, so not yet in patients. This is Hunter syndrome, Gaucher type 3, which is the neurological version of Gaucher, and Pompe disease. Next slide, please. So gene therapy is a very, very broad category and can be very confusing as the term is used very generally. And there are really 3 ways it can be done. You can edit the actual gene. So basically gene therapy, the purpose is where someone has a variant of the gene, which maybe produces a protein which is abnormal and therefore doesn't function as well as it should, and as a result causes a disease. So you can correct the gene, a bit like word processing, go in, change the spelling of a word, and that's gene editing. We hear a lot about that with CRISPR. Then there's gene addition and gene augmentation. So gene addition and gene augmentation are very similar in many ways. What we're doing here is the copy of the gene that the person had in the first place stays as it is, but we put in a copy of the gene that codes for the normal functional protein. So it's an addition. And that's what I'm going to talk today about, how we add additions. And that's true for the therapies I'll talk about. And also we're going to hear from a patient who had CAR T therapy, and that is also gene addition, where an extra gene is popped back in. Go to the next slide, please. So how does gene therapy work? So what it requires is that we need a copy of a functioning gene to make the protein that we need. And the gene has an error in it. So it doesn't read quite right. And as a result, the protein it produces doesn't function as well as it should. And that impacts the ways the cells in our body functions, and it therefore impacts all our tissues and organs. And so there's really 2 components to gene therapy. One is we have to put in the correct genetic information, so the right protein can be made that functions, but we also have to have a means of getting it in. Think about it like a letter and an envelope. The letter is the message. It contains the code so that the protein is produced in the correct manner. And the envelope is very much so that it can be targeted to where we want it to go. And we call that a vector. And very commonly, They're viruses that we use. So those modified viruses, we call them viral vectors. They're no longer viruses. Viruses, by definition, would have to infect and then be able to basically reproduce and make more virus and then infect again, just the same as we all know with COVID-19. So this starts out based upon virus principles, but we just take the components we want. We just want the delivery components, not the pathological disease-forming components. We just want its ability to be able to deliver genetic information to cells. And therefore, we take basically a few components out of a virus to make our vector, add in the contents, the letter effectively with the code, and then that's what goes into the cells. And there are two basic approaches. There's lentiviral vectors, which I'll discuss in more detail, and AAV vectors. AAV vectors work slightly differently in that you put the vector directly into the patient, with ours we'll take the cells out, and I'll come back to that in a few minutes. Next slide, please. So this is the two approaches. On the left-hand side, you see where the vector with the code is popped straight into the patient, given normally intravenously, or can be injected directly into an organ, and then basically travels or is already in the cells it needs to go to, so that the genetic information can be provided to those cells to make the correct protein. And quite often it's given intravenously and it travels to the liver, and the liver then makes the protein. And that's one approach. The approach I'm going to discuss today is the right-hand side, so-called ex vivo. What does that mean? Well, it means we take cells from the person, modify them outside the body, so the viral vector never goes into the patient, and then we pop the cells back in. So they're delivered via the cells from the patient themselves. Go to the next slide. So I'm now going to just tell you a little bit more about that second process, because that is what AvroBio does. So if we look at position one, we have to get some cells from the person. And the way we do that is we use a drug that's been tried and tested for many years, or two drugs, I should say, GCSF and Pyrexafor. They're basically two drugs that are used all the time in bone marrow transplant. They're used to basically mobilize stem cells, the cells that produce all the blood in our body, from the bone marrow into the blood. And once they're in the blood, we can capture them. And we basically take off a person's blood, pop it through a machine, and take out the cells that are of interest to us, the so-called stem cells, and return the rest of the patients. That takes about 4 hours. Having got the so-called CD34 cells, that just means a stem cell. That's a cell basically that can make all the different blood types. We then modify them with the lentiviral vector, which I'll come back to, so that there is basically an additional copy of the gene. We've now put in a gene which codes for the therapeutic protein that we need. And that just takes something like 3 days. We then freeze down the patient cells, which have been modified with our gene, and then we test them. It takes about 6 to 8 weeks prior to them going back to the patient. They literally just infuse. We just pop them into a vein, and then they home to the bone marrow where they always lived, and then they produce all the patient's blood. And because they do that, those cells, which have a copy of our gene in, will therefore have that protein produced in those cells, which either corrects those cells or enables protein to be released into the body where it can be picked up by other cells. Think there's little factories that produce ERT. Next slide, please. And there's some very special things about ex vivo gene therapy in that we can truly target the whole body, head to toe. And by that, we mean brain as well as body. And that's very important because when we think of enzyme replacement therapies, because they're a protein, if you put them in intravenously, they can't cross the blood-brain barrier. And therefore, you can't really impact the central nervous system, which is very common in lysosomal disorders, to have a contributory factor to the overall pathology of the disease. Now, it's not obvious, but the stem cells in our bone marrow produce all the blood. And that's what you see on the bottom of this figure. And we all know that the stem cells produce our blood, all the different types, and therefore all those cells, those trillions of cells, produce our therapeutic protein because they've all got a copy of that gene. But very importantly, one of the cell types that's produced from these CD34 cells are called monocytes. And monocytes are very special because they can travel to the brain and go across the blood-brain barrier and into the brain itself. And because they have our gene, all their daughter cells, so-called microglial cells, which are throughout the brain, you see them in the figure, the round figure on the right-hand side. So all the microglial cells in the brain will produce our protein and either secrete it or correct the microglial cells. And that's very important for lysosomal disorders that have a CNS component. So we have a true head-to-body approach to treating disease. We go to the next slide, please. Now, to enable that, we need a very special agent, which is called busulfan. Now, it's called conditioning. So what is it? Well, conditioning, it's basically a means of making space in the bone marrow. Unfortunately, when we take the cells from the bone marrow then fills back up again. And then there's no space for our genetically modified cells to go when we put them back in again. And that would be no good. And people did try it and it just didn't work because the cells had nowhere to go. So we use an agent called busulfan. And busulfan has a number of special properties, but one of the really important properties is that it enables the cells to get into the brain. Those monocytes I talked about going across the blood-brain barrier to form microglia, because that way we can offer potentially a head-to-toe solution. The drug has actually been around actually about 70 years, invented in the UK originally, and it's given over 4 days, and we monitor it very carefully. We take blood samples regularly after we've given it, and then adjust the next day's dose, because it's a bit of variability between people. It's important to say that it is part of a regime. It was originally invented to treat cancer back in the 1950s, but has since had many years of patient and clinician experience. And so it's a well-known drug in terms of its side effect profile. But it's important to note that it does have potential side effects. And you really must, anybody thinking about coming on a clinical trial must discuss them with their clinician so they can really understand the risks and the benefits. Next slide, please. Yeah. So as I said, we've enrolled 16 patients in our first 3 studies. The Fabry study, which was actually kicked off in Canada, and I'll come back to talking to you about that shortly, is the one we've treated the most patients on. Gaucher, we treated our very first patient, and cystinosis, there's 3 patients treated, and the other 3, as I said, are still in preclinical studies at the moment. Next slide, please. So this is our Fabry disease. This is our first gene therapy clinical trial. The phase 1, so that's the sort of safety and early efficacy phase, was actually done in Canada, and we're very proud of that. We still actually have offices in Toronto as well as in Cambridge, Massachusetts. And this was a study which looked at 5 patients who were all on ERT prior to the start of the gene therapy, and they were aged 18 to 59. And you're going to hear from one of the patients shortly. And the interesting thing here was that originally this was really a sort of safety study. So the patients were on ERT. And after they'd had their gene therapy, they went back on. So it was a short period where they were taken off ERT and then they went back on again. And some of the patients did so well that the investigator went back to the regulators in Canada to ask if the clinicians, if it was appropriate, could actually take the patients off their ERT. And I'm sure that Darren will talk about that. And something like 3 out of the 5 patients are now off their ERT. The phase 2 is quite different. So this is patients who've never been on ERT. This is actually an AVROBIO study. As you can see, it's in 3 different countries, looking to enroll 8 to 12 patients, and we've dosed 7 patients so far, 16 to 50-year-old males. And again, as I said, these are patients not on ERT. So very different to the first study. So here we're looking really across the 2 at patients who've been on ERT and patients who've not been on ERT. Next slide, please. Our Gaucher study is for Gaucher type 1. So this is where patients do not have a neurological component. We are looking at Gaucher type 3 later on, and we are doing preclinical work on that now, but that's not yet in the clinic. So this is for patients who do not have the neurological complications of Gaucher type 3. It's a safety and efficacy study, and the goal is to enroll something like 8 to 16 patients aged 18 to 45, male and female. We need to make sure that patients have got a diagnosis of Gaucher type 1. And that's challenging because of different mutations and different interpretations, but basically without a CNS component. And they must have been stable on ERT for greater than 24 months, 2 years, or ERT naive, because there's 2 arms to this study. And we are now currently recruiting this trial. Next slide, please. And our cystinosis, this is being run by, as an investigator-sponsored study at University of California at San Diego, phase 1/2. 3 patients actually treated to date out of this target 6 patients. They're both adults and adolescents, and there are 2 cohorts, but greater than 18 and greater than 14 in the final cohort, male and female. And this is really, again, is a safety and efficacy study to look at the gene therapy. And this is quite different. So as people may appreciate, the first 2 I talked about were diseases where the enzymes don't function correctly. This is a transporter protein in the actual lysosome called cystinosin. So slightly different, but still the same approach. Take cells out, modify them, put them back. Next slide, please. And then we have 3 programs which are in the final stages of preclinical development. Hunter, which we licensed out of the UK, out of the University of Manchester. Gaucher disease type 3, which is basically almost an extension of our Gaucher type 1, but this time for patients with neurological symptoms. And as I explained earlier, we would expect that CNS component because of the use of busulfan and the ex vivo lentiviral gene therapy with the monocytes crossing the blood-brain barrier, and POMPA, which is truly a CNS and muscular disease combined, so really would benefit potentially from head-to-toe therapy. Next slide. So in summary, we're developing a range of lentiviral gene therapies for lysosomal disorders with really the potential to really free people from a lifetime of genetic disease, not requiring infusions every 2 weeks for enzyme replacement therapy. It is an approach where we take the stem cells, we talked about these blood-forming ones, CD34 cells, we modify them outside the body, that's why we use this term ex vivo, outside the body, and then we introduce them back into the patient. We just infuse them in, where they home back to the bone marrow, and they produce blood cells which go around the body, and they release the protein like mini ERT factories, basically traveling throughout the body, releasing the protein of interest. And also some of them, the monocytes, cross the blood-brain barrier into the brain, where they form microglial cells, which again will produce the protein and either correct those microglial cells or release the protein into the brain, where they can correct other cells. The conditioning with busulfan is a very important part of the therapy, because we really do have to make space to enable these cells to go back and therefore to home to the bone marrow and then engraft there for the life of the patient. And the busulfan also has the advantage in allowing this conditioning of the brain as well, so we can get our cells into the CNS where they can function. We're currently evaluating safety and efficacy in 3 clinical trials: Fabry disease, Gaucher disease type 1, and cystinosis. And more information can be found about all our clinical trials on clinicaltrials.gov. And I'm very happy to take any questions that people may have.
Sorry, take this back in. Okay, that was an amazing, very, very cogent, but also very comprehensive overview. I don't see any questions in the chat box, but again, if anybody has any questions for Chris, we're happy to take them now. I didn't know whether you're going to stay through Darren and Brian's presentation as well, but that's up to you.
Yes, I almost certainly will. I mean, that's my plan, to stay on.
Okay.
So, I always learn such a lot from people, and especially from patients who have the therapy.
Well, now we've got questions, so we're going to get you going on here.
Yeah, I can see the
question
flashing
here that
in.
says, what about side effects so far? And I think that is, as you emphasize, something really important for patients to ask about. And we'll hear a little bit from the patients themselves, but in your experience now with the range of patients that you've had.
Yeah, so first of all, I must say that safety is number one. I mean, that's something that's super important to everybody at AvroBio, something we think about a lot. And so you can divide safety up into 2 broad areas that people have concerns about. There's obviously concerns that, you know, we're using a viral vector, something based on a virus. Can you, I mean, you know, is that going to cause challenges? Well, what we can say is that it is based on HIV. It takes the components, the delivery components of HIV, and uses them like an envelope to deliver the gene of interest. And something like 350 plus patients have been treated to date using lentiviral vectors for monoinherited genetic disorders, such as lysosomal storage disorders, thalassemia, sickle cell, and a variety of other indications caused by single gene defects. And to date, there has been no challenges with the vector. Likewise, thousands of patients have been treated using lentiviral vector to modify T cells to make CAR Ts, similar story. So the safety profile to date for the actual vector incorporating a gene into cells is very good. And I should just say that, you know, HIV has infected something like 70 million people around the world, unfortunately. And if you look at the challenges for HIV patients, again, the virus, in case it is a virus this time with HIV, integrates into the patient's cells. That's why, unfortunately, if you catch HIV, it's there for life. And what we can say is that the integration of that vector is very similar, or that virus is very similar to our vector. And again, I do not know of a single patient who's come to harm because of the integration site of the vector in those 70 million patients. So, you know, the vector has a good track record, is what I'd like to say. Then the second part that we will think about is the conditioning, the busulfan conditioning. Darren will talk about a different conditioning regime that he actually underwent. So for the two clinical trials, we've actually used two approaches. Melphalan was the original one and busulfan was another. They're well-known agents used to make space in the bone marrow compartment. Now, sometimes it's easy to get confused because they are used in oncology. In the oncology setting, they're used, in fact, typically in cocktails with multiple other drugs, because the goal there is to really hit the cancer cells very hard and eradicate them. You need to use them to cure the disease. You're there, so you really hit very, very hard with combinations and cocktails, often several times, to try and remove a cancer. So very hard. And typically, patients have received multiple lines of therapy before they actually get to a transplant requiring the same sort of drugs. So, you know, they've had a hard road before they even got there. And so, sure, there are complications there. But this is very different. We're using conditioning in a very different manner. We use it as a single agent, single cycle, so just 4 days, very tightly controlled to a very specific exposure. And as I said, it's just one agent. So we're not talking about multiple different agents in patients who are far fitter, far weller. And so I think there's an important distinguishing feature there as well about the use of the conditioning agent. And the benefit of the conditioning agent is that it allows true head to toe impact of the cells. Because, you know, when we think about lysosomal disorders, you know, they're not just confined below the CNS. Unfortunately, the genetic mutation is throughout all the cells in the body, and therefore, really to have a lifetime impact, we believe you need that head-to-toe effect.
Well, that was an amazingly comprehensive answer to the.
I don't know if it's you. I think, Darren, you might be on mute.
I am indeed. Sorry, I didn't want to interrupt you when you were speaking. You've got a whole host of wonderful questions here. And I actually had a bunch of them myself, but I'll save them for later. Is your technology applicable to muscular dystrophies? I know this is one of the areas that people are really keen on in terms of not other therapies available.
Yeah, so I have to be truthful. We have not looked at that, but gene therapy is being used in that space, and there's some very nice early clinical trial data. So I think it's one of these areas of watch this space, but it's not an area which AvroBio is into, because it's not, again, we're focusing on our technology. Our platform is take out stem cells, modify them, put them back. And you can't really do that with Duchenne muscular dystrophy. You've really got to work on the muscle. Got to it's the AAV in vivo approach where you actually put the vector itself in. So I think there's some really nice work going on, but it's not what we as a company are doing. But I think it's one to watch this space, because I think there's been some really great work in that area.
Yeah, so it's a really different purpose, eh? As
Very
what,
different. Yeah.
yeah,
Yeah.
yeah.
I mean, I think we have to say that, gene therapy can be done in lots of different ways. You've got to pick the appropriate gene therapy for the disease indication. And if you want head to toe of a protein distribution, ex vivo lentiviral is a great way to go. If you specifically want to target just muscle, then AAV may be a good way to go. And POMPA, people say, well, it's a muscle disease. Well, POMPA is actually a disease of CNS and muscle. And therefore, we believe that our approach, our head-to-toe approach, again, is very valuable there.
Okay.
I'd like to pick up, because I know time is tight, but I would like to pick up the antibody one, because I think that's really important to people, and I really do want to talk about that. So everybody is aware, I'm sure, of your organization that, if you give a protein and give it every 2 weeks, unfortunately, it's a bit like vaccination, a bit like giving COVID vaccine. You raise an antibody response. You give the protein the first time, great, the body sees it, but unfortunately, then the antibodies and things can kick in. And then when you give the protein a second time, and then you basically reinforce that immune response, and that's really unfortunate, because then when the drug goes in subsequently, the antibodies attach to the drug and reduce its effectiveness, which is not what we want at all. The gene therapy approaches have a big advantage in that those cells which I talked about, which we take out, I said they produce all the different blood cell types in the body, including things called antigen presenting cells. What are they? Well, they're cells in our bodies which allow our bodies to recognize ourselves as ourselves. Otherwise, our immune system would attack us. And so what happens is, because those cells actually make the protein, the correct protein, they enable autotolerance. And we've seen that in other clinical studies. It was seen in a similar ex vivo lentiviral gene therapy in Italy, where they were looking at Hurler's patients, and 4 of them had very high ERT antibodies, which after 1 to 2 months were reduced to 0. They couldn't detect them anymore. And we've actually seen it in our own clinical trial, small levels of antibodies, lower levels, again, reduced to undetectable amounts. So it's inherent in the approach is that we can create this autotolerance, and that's what sets us apart. And so even if you have high antibodies coming into the clinical trial, it's likely that we will be able to get tolerance against those. But also, very importantly, our gene therapy won't produce an immune response because of the nature of the way we deliver it, and these antigen-presenting cells enabling tolerance. Basically the same way as you and I have our own system, so that our immune system doesn't attack us.
That's amazing. So why don't, if you are willing, Chris, this would be so amazing, a benefit. Why don't we go to Brian and Darren, and they've got a conversation. And as I said, I think you'll find it interesting. And I'm sure everybody will. And you'll hear some of the questions that came up here in their conversation. So I think that'd be great. So Angela, I'm going to turn it over to you, and with huge thanks there. Hello and welcome back. Thank you so much for the presentations around the lentiviral vectors. And what we're going to have now is an opportunity to talk to 2 individuals who actually had the opportunity to have access to therapies that are based on that technology. 2 amazing gentlemen, and we'll have a good chance for them to tell their stories, a bit about how they got into that decision, and then what has been the outcomes of it. But first of all, let me just ask them to introduce themselves. I'll have them introduce themselves, and then we'll have a bit of a conversation with them.
I'll go first. Hi, I'm Dr. Brian Kaufman. I'm a retired family physician. I was actually born in Canada and practiced in Canada and the United States. And I have chronic lymphocytic leukemia, CLL, and that was difficult to treat, and ended up getting a biological treatment that used a lentivirus to help control it, and that's what we're going to be talking about. So during that process, I also founded a nonprofit, the CLL Society, to help other people gain access to the latest in clinical trials and research.
Hello, my name is Darren Badalka. I'm 52 years old and have Fabry disease, which I was diagnosed with at about 36. I live in Vancouver, BC, and I did a gene therapy trial in 2017.
Thank you, folks, very much. What I'm going to do is ask each one of you maybe to kind of talk a little about how you came to get the lymphviral therapy that you did, how it actually worked for you, and where you are now. And I'm going to leave it as a conversation between the two of you. So feel free to interrupt each other and to kind of build on what the other one is saying. And at that point, I'm going to excuse myself and turn it over to you guys.
Great. Well, Darren, I'm very interested because you have, I thought I had a rare disease, but you have like a really rare disease, and which has some good treatment options, but not a whole plethora. So I'd be very interested in your story a little bit, and then I can tell mine. And because in cancer, there's, at least in my cancer, there was always different options. So there was a bunch of choices, and I think yours was my either do it or don't do it kind of choice, I think. So, yeah. But maybe I
to
got
some
that
degree.
wrong. Yeah.
Yeah, well, sure. I'll tell my story. So, diagnosed with Fabry disease in 2005. Thankfully, at that point, there was treatment available. So basically, the condition of Fabry disease is you are either deficient or absent a particular enzyme. So the treatment is basically to get that enzyme. So a manufactured version became available in sort of the early 2000s. I went on to that treatment right away. And interesting for such a rare disease, there are actually more and more treatments becoming available. I think the one that we were all, as patients, very keen to see the development of was gene therapy, because it's the one that, so many of us sort of hang our hats on, is like, could this be the thing that, if you're diagnosed young enough, that really could avoid a lot of the problems of Fabry disease? Like most of these conditions, once the horse is out of the barn, so to speak, and damage is done, damage is done. So at my age, there's damage, but there's that real hope in the community that something like a gene therapy for someone young enough may mean that they don't experience the damage.
So, and I'm going to ask a question, and
Sure.
I hope that's all right. When you got your gene therapy, this was not a standard of care by any means. There's a little bit,
Oh, no.
You're being a little underselling your sort of casualness about jumping into gene therapy. Could you tell us a little bit about that experience before I talk about mine? Because I think it's pretty dramatic. Yeah.
Certainly. Well, like there's parts that are dramatic, and I think parts that are less dramatic. And I'm a fairly sort of clinical kind of person in how I analyze things. So the decision I had was whether to basically be the first person treated for Fabry disease with gene therapy. But I broke it down at the time into kind of different sort of pieces to decide whether that was the right choice for me or not. Is while it was the first trial of gene therapy for Fabry disease, it certainly, many of the aspects were fairly common. Like, interestingly enough, given your condition, like a lot of my treatment actually occurred in the cancer ward. So a lot of the technology for it was basically the same as what a blood or bone marrow cancer patient would go through when they have a stem cell transplant. So the basic process, getting just straight into the actual study, was they collected stem cells through a process called apheresis. So basically you are connected to a machine where they harvest your CD34 stem cells. Those cells get reprogrammed, and eventually you do a round of chemotherapy to basically kill off a certain amount of your bone marrow to give those reprogrammed cells somewhere to go back and live. And assuming that transplant is successful, then the goal is that the body would start to produce that deficient enzyme, which is what it did.
So maybe I'll jump in there and tell a little bit of my story. I was diagnosed with chronic lymphocytic leukemia, which is a chronic cancer, the most common adult form of leukemia in North America, but still quite a rare disease. And when I was 54, and it comes in many different flavors. And some people have a very slow-growing indolent disease, never need any treatment. But I wasn't one of those lucky ones. I had certain markers that suggested my disease was going to be aggressive, and it behaved very aggressively, as a matter of fact. And within the first couple of years, I had multiple hospitalizations, emergency removal of my spleen, internal bleeding. And at that time, and this was 16 years ago, there weren't any great treatments. And I went on to go to have a bone marrow transplant early on, because that was the only potentially curative therapy for me, which didn't work. But it had a lot of the same things. You get some chemotherapy, not so much to control the cancer, but to clean out the bone marrow, to make space for the imported immune system, the new blood system that I was importing, that would take over and kill off any remnants of my cancerous old blood system. Fortunately, it didn't work. And I think one thing that you and I share is that we're early adopters. So I jumped in, not into CAR T therapy, which I'll talk about later, chimeric antigen receptor T cell therapy, which is the lentivirus therapy, but into a targeted therapy that was brand new, a small molecule that was orally absorbed, so a pill, in a phase 1 trial, which is like a first group, one of the first few hundred people in the world. And it revolutionized the care of the kind of leukemia I had. And I got about 7 years out of that therapy, a drug that only had a number when I started, PCI through to 765, which later became ibrutinib. But why I point that out is a lot of this has to do with timing, because if I hadn't had the transplant, if the transplant hadn't bought me some time, if that hadn't gotten me to the next clinical trial, then that clinical trial got me to where I got 7 years out of that. I was starting to relapse. And then I had to look at other options. And by this point, when I was relapsing, my cancer outsmarted this drug, had what's called a gain of function mutation, and it found ways to work past what the drug was blocking. So I was in a lot of trouble. And there was a buzz about CAR T therapy, this chimeric antigen receptor T cell therapy. Still completely experimental, in CLL. But thank goodness it's now approved for some other types of lymphomas and childhood leukemias and young adult leukemias, because it's so darn effective. But like a lot of therapies in cancer, where these things are first used is kind of at the end of the game, where people are running out of options. And one of the things I hope we get time to talk about is how we can move these things up sooner, so you don't have to wait until you're damaged by a whole bunch of rounds of chemo and other therapies, and get it in sooner. So I looked, I still had a couple other options, a couple other clinical trials. But what excited me about this was just the science behind it. And like you said, what would happen here is that I would go through leukapheresis, which is what you're describing. They would they would take out my T cells, which are a type of lymphocyte, a type of white blood cell, and infect them with a lentivirus that had been, the virus itself had been genetically modified to recognize my cancer cells. And they would grow them outside of me, and then they would transfuse them back into me, and where they'd go to sleep for a little while and then wake up and say, oh my goodness, look at all these cancerous cells around. And then there would be a killing frenzy. And the trick with that killing frenzy is there's a lot of collateral damage, and I had quite a bit of collateral damage. That's called cytokine storm or cytokine release syndrome, which has got a lot of press recently in relationship to COVID. It's the exact same thing. Cytokine is just a fancy way of saying inflammatory enzymes. And my inflammatory enzymes were thousands of times higher than they should have been. And I felt it with like the flu from hell, and above and beyond. And also had what they euphemistically call neuro events, which is kind of a toxic neurological state where I was confused, word salad, didn't know what was going on. I had to learn how to walk again in horrible pain. But I got through all of that, and there's drugs that block those inflammatory responses. And when they restaged me a month later, and they did a bone marrow biopsy and my blood counts down to 1 in a million cells using the genetic signature of my cancer, they couldn't find the cancer anywhere. And all my lymph nodes had shrunk back to normal. So that doesn't mean the cancer is gone, doesn't mean it's cured, but 3 years later, I'm still in that same condition with no progression of the lymph nodes, and the cancer at 1 in a million or less in my bone marrow and blood. So it's been nothing short of a miracle, but it was a high risk situation, but I decided to go for it because I just loved the idea of a living drug that would stay alive and kind of play whack-a-mole with my cancer as it
Yes.
popped up again. So uh, it was a exciting, thrilling time. I just remember the reaction my wife and I had when we got the news that there was no cancer to be found. I mean, that's what
Yeah.
we'd gone. And I had to move to Seattle to do this. I live in Southern California. We had to move up there for a couple months. We lived in a hotel, had to walk to the cancer center when I wasn't in hospital. It was a big change, a big stress, but boy, the payoff was amazing.
So both of you, let me interject for a second. So both of you had what seemed like a very successful experience with it, but obviously a big challenge. How did, let me go back to you talking about your wife, and I know Lisa, Darren's wife, well, so yeah, how did your spouses take this? What did you explain to them? What was the discussion? Because there's still a high risk. You're really, putting yourself in a high risk situation when you're actually undergoing this. And sounds like, Brian, you didn't have a whole lot of other choices because you come to the end of a string of it. But Darren, you actually could just have continued with the replacement therapy and even some newer versions of that replacement therapy. How do your family and especially your spouses feel about this?
Sure, I'll jump in. So my wife is very familiar with my condition and as well as the treatment options. I had I'd say a few real benefits going for me in terms of my comfort around the study. One was I knew the lead researcher for over a decade when I did it because I was quite involved in patient advocacy for a period of time, and I knew many of the other people on that research team. So that increased my confidence, and Lisa knew a number of them as well. So she had a confidence in it. I participated by donating stem cells to the research and had a sense that they were quite successful with the treatment at that research stage with my cells. So that played a part of the decision making. Then basically Lisa and I sat down and we went through all the different risks and decided, could we mitigate them? Could we do our best to sort of reduce the probability of that risk occurring? And we came to the conclusion that we could. And then on the flip side, we considered what are the risks of not doing this procedure? And one of the key things with Fabry disease is that the blood-brain barrier doesn't allow the enzyme replacement therapy to make it to the brain. So there's some real questions around the stroke risk, even if you are on ERT. So that played into our decision making, and ultimately we considered all of it as a package and decided that the right choice was to do it.
And there's remarkable parallels in that decision making too, because I think one of the things that you point out so clearly is there's a risk in not doing anything. There's significant risk.
There is.
And I did have other options, but sometimes there's a window of opportunity. This was a trial that was open. The trial was open to 40 people. I was patient 36. It was really a sweet moment because they'd gotten the dosing figured out in the first 30 something patients. So they knew what the right dose was. They found, just the right, bowl of porridge for Goldilocks,
Yes.
right there. And there had been one fatality in the first 36. So I knew it wasn't without risk, but it wasn't a huge risk in cancer treatment at this stage. A 3% risk is relatively low of mortality. So I knew that. But getting to Doreen's question about my wife, with CAR T therapy, because of the effects on your cognitive function and your vital signs, you're incredibly dependent on having a caregiver. And in fact, most places will not allow patients to have CAR T therapy without a caregiver. And for most people, that's our spouse, our significant other, but sometimes it's an adult child, sometimes it's a parent, sometimes it's a best friend, sometimes it's a professional nurse, but you need that because to give you an example, I was completely out of it. I went and I was trying to sit down and I was in such pain, I couldn't sit down. and it was taking me 15, 20 minutes. And I was arguing with my wife saying, what's normal? It always takes me 20 minutes to sit down. I don't know why,
Yeah.
and, she says, I'm not listening to you. I'm calling the docs, and the docs called and they sent the ambulance over. And next thing I was on a stretcher and going to the hospital, and thank God that she was there, because if I trust my judgment, which was altered at that time, I was running fevers, I wasn't making good decisions. And when you're out of it, you're kind of out of it, and you don't even know that you're out of it. So it was my wife who had to watch me. And for her, she was the one who's saying, am I going to get my husband back? Is he going to walk again normally? Is he going to talk again normally? My adult children came up to support my wife, and I was completely gone. I had conversations with people that I don't remember at all. I was completely confused. And Patty had to take over and make those decisions making. But we'd had talks before, and she knew what my wishes were and where we were at. And I'll just share one little story. The head of the research came in from Seattle Cancer Care Alliance, came in and said to my wife, he came visit us at the hotel the day before this was all starting, and said, I just want to tell you, Patty, and he turned and said, Brian, I'm not talking to you, I'm talking to Patty, that no matter how bad things get, don't pull the plug on Brian. This is kind of a strange message to get.
Yes.
Don't pull on the plug. I can get him back. It may look terrible, but don't pull the plug. I can get him back, which was kind of an interesting message to get. But in one ways, it prepped us for saying, he could get really bad, but he'd still come back again. And that's kind of what happened.
So let me interject again, because that's one of the things that, we're talking about, both of these still in experimental stages. And Darren, I don't understand why, it's been like 5 years. So you would think that, by this time they could have this, stuff out there available, but obviously it's a long research process. But what if both of you seem to indicate that, okay, there is in fact a mitigation strategy. We can reduce that risk, I think is what you said, Darren, and,
Yes.
What you're saying as well, Brian. How important is it if you were giving advice to another patient, which I'm sure you get a lot of requests for, and I know, Darren, a lot of patients, but a number of patients in Canada have actually taken the therapy because of you, the gene therapy. What do you say to people when they say, what is the worst possible scenario, and and what is that mitigation strategy?
Sure. Well, I say I'm not a medical person by training, so I would direct the person back to the disclosure statement and understanding their consent, because I don't want to give anybody medical advice. So I would just tell them what it was I did, which was, it seemed to me that the key thing that one needed to be worried about was the fact that after the chemo, there's a big reduction in your white blood cell count, which is basically your immune system, and that you are quite susceptible to getting sick. And if you do get sick, you don't really have the immune system to fight it off. So my advice kept coming back around to, well, take this seriously, take the advice of the medical professionals, be very, very careful about getting sick and exposing yourself to anything where you might get sick, not only during the time where you've had the chemo, but in the time like leading up to it. Because if you got sick, you couldn't get the chemo. They're not going to give you chemo if you're already ill. So, ironically, this set me up well for COVID. Like, I was quite practiced at staying away from people and not exposing myself to anything where I might catch a flu or a cold. So that to me was really the key risk. Don't get sick. So take some precautions, take some amount of your freedoms, park them for a short period of time, because this is a long game. Like, if I can be making enzyme and reduce my risk of stroke, I'm willing to do a tremendous number of things that cause a little bit of short-term pain. Like, certainly not seeing friends and family for a little while, it's not the greatest. I'm fairly social, but hey, long-term, right choice.
Yeah, and I would add to that, when you're with what I was doing, it was a different set of risks, and that it was experimental, but the risks, you have to compare the risks to the risks of not doing anything, and the risks of not doing anything was that I really had one or two other options left, and they would likely be short duration, and then the CAR T might not be available because it was experimental at that time. And I kind of did sort of a heart thing, saying, if what would happen if I couldn't get the CAR T? How would I feel versus getting it? And there was
Mhm.
trepidation in both
Yeah.
settings, but the fear was greater. Oh my God, what if I can't get it? You know, I mean, because I saw it as a chance, as a way out of this dilemma. And the big fear I think with gene therapy is you kind of live with is the unknown, because at least in my trial, the National Institutes of Health is following me for 25 years after. I mean, they draw blood on me. And follow isn't, you know, tightly. I mean, a blood test once a year is essentially what I do, is send in. But that tells you that we really don't know. You know, I mean, there's always in the back of your head, is there something that's going to pop up 15 years from now that you don't know about? But I knew what was going to pop up in 15 years if I didn't do that, and that was the
Yes.
cancer, and probably a lot sooner than that. So it just seemed like this was worth doing, and all the risks were, you know, really quite manageable. I just, one thing I want to add: your prep for COVID is amazing, because cancer patients actually have a lower incidence of COVID, because we don't, if somebody's coughing and sneezing, we were crossing the road 2 years ago. You
Yes.
know, I was wearing a mask on planes for the last, you know, 5 years, you know, when my immune system was corrupted, you know, by the cancer. So
So that's an amazing, we're almost out of time, though. I could probably listen to the two of you chat for, you know, many hours after this, and maybe we'll have to figure out how we can make this into a true documentary. But beyond that, a lot of people talk about these therapies as curative. I mean, we know that certainly CAR T therapies, certainly for children with leukemia, have been very long lasting. We think about, you know, gene therapies as, you know, people talk about them, and we sometimes back up and we talk about them as, you know, forever therapies. We talk about as durable therapies. What is your sense of it in terms of what's your hope for this? And what would you, I mean, I'm going to ask you a double question because we are running out of time. What is your, in your own mind, what do you think about it in terms of the lasting nature of this therapy? What do you hope for it? And secondly, what do you hope will happen in terms of getting this therapy available for other people?
Well, I seem to go first, so I'll jump in. Uh, I, when I did this, I had really no expectations. I mean, this was phase 1 safety study. So certainly my goal was don't be permanently harmed by it. And I didn't think that was going to happen. The fact that I started to produce enzyme at a level where I no longer take ERT is just miraculous to me. So as at my age, there is a certain amount of damage done with Fabry disease. That's just the way it goes. It's not going to reverse. But if I can stay stable and have a slower progression, again, that is a absolutely wonderful result. Where do I hope this goes? I hope that this can become a standard of care for people where it makes sense for them. Like, probably not every person that has Fabry disease needs to have gene therapy if their disease is mild enough. But for those where it makes sense, I certainly hope they get access to it. And my even bigger hope is that this sort of methodology of treating people can be extended to many, many different single gene defect conditions.
doctors are always hesitant to use the C word, cure, and in CLL, chronic lymphocytic leukemia, the paradigm of treatment is you do one therapy, kick the can down the road, and it gives you a few years, and then you do the next and the next. And what enticed me in this is the possibility of kicking the can over the fence and being done with it. And nobody's going to say that, but there are people that are more than 10 years out that have no evidence of disease. And so can it drive the cancer down to such a low level or wipe out every single cancer cell? We don't know the answer to that, but we do have the hope that that could happen. And for me, that was a really exciting option. So, you know, I don't think of myself as cured, but I think of myself in a very durable remission, and I'm hoping that I'm cured, and I think that's a reasonable choice. To get it to other people, you know, people have to enroll in clinical trials. They have to be aware. That's the only way you get this over the finish line. That's why we set up the nonprofit CLL Society to let people know about these, and these are available in other disease states, and bluntly, you have to do a lot of advocacy. We work to make sure insurance pays for this because this is a very expensive therapy. So there's a lot of pieces that we do. And I wanted to make sure that you didn't have to be like Darren or myself, an advocate, well connected, know the researchers, that everybody should have the benefit of these kinds of therapies if it was appropriate for their treatment.
Well, I really can't thank both of you enough for your really amazing stories, for your insights in terms of, coming into the therapy, but what that therapy really means. And also, as you say, what that hope is for the future. It's a great spot to, I think, end this interview. But hopefully we may be able to come back to it and really talk about it in terms of those next stages. I think what we are going to be talking with many of our participants about is the big challenge, and that is, how do we get it not only beyond the clinical trial stage, but if it becomes available, how do we make it available to many other people? And especially when we talk about, people that are in, as you're saying, not necessarily as well versed or connected, but also people that live in very challenged economic environments, where, I mean, this is not a cheap therapy. It's not going to be a cheap therapy for a long time, but that should not preclude it being made available to people who are living in, healthcare systems, or even in countries where in fact there is not going to be the same level of support. So that's also something that's very important to us in terms of these therapies. So huge thanks to you, Brian, and to you, Darren, for taking the time to share your stories. And also, though, huge thank you for remaining advocates and really taking the opportunity to help educate others, but also to making sure that these therapies become more available. We'll look forward to your next chapter. Thank you so much.
Excellent. Thank you very much.
Okay, so that was a most informative discussion. And again, certainly two pioneers in the area, but also people who bring in a great deal of information. I'm wondering if there is, there's a few other questions, and maybe if you don't mind, I can ask you, Chris, to come back on and we'll answer a few more questions. Then we'll talk a little bit about where we want to go in global access. Is that okay? Or maybe I'll just ask you off the top. Can you give some reflections on the conversation between Dennis and
fantastic.
Brian? Yeah.
Yeah, so I thought it was really good. I mean, I think it's always good to hear people's journeys, and I think they describe them really appropriately. And we heard Dr. Kaufman have quite a challenge with his therapy because the nature of cancer treatment. And it was what I was trying to say earlier, the need for eradication. And you really got to throw everything at it if you want to get the result that we all desire, that long-term, or as he described it beautifully, kicking the can over the fence. That was wonderful. Very different with the mono-inherited diseases, where we're just trying to put a little bit of enzyme back. We need nowhere near that level of aggression to really kill off all the cancer cells. We just want to create a little bit of space. So very different. And I think that was very encouraging to hear Darren talk about two key aspects for me. One is the CNS, which he valued very highly, the potential that his ERT wasn't going to help him with the future events, CNS events. But also, I'd just like to draw out one thing. But Darren had the original conditioning agent, which was melphalan, and he very rightly described the fact that the B and T cells are significantly knocked down by the melphalan. And it was one of the reasons we switched from melphalan to busulfan, because that has negligible impact on the Bs and the T cells. So Darren very rightly talked about being very careful for months afterwards while his Bs and T cells recovered. And that was one of the reasons we switched to busulfan, to not just enable us to really target the CNS, but also to make sure we didn't impact people's immune systems. So yeah, I enjoyed the two talks. They were fantastic, weren't they? They were inspirational.
Well, there were a number of other questions. I don't know if there's anything that has not been covered. I think there was a question about benefit risk ratio, but and there were some additional viruses. I think we had some, you spoke to that already, sort of where the, the benefits for, or the appropriateness of certain types of, the AAV viruses versus the lentiviruses versus, maybe some other technologies, but
yeah, the non-viral approaches. Agreed.
yeah, exactly. So I think we did have the very first discussion we had was talking about zinc fingers. So we've kind of covered a good range of we should make sure we do a small quiz to all of our participants and see if people have been able to pick up on these nuances, but really that's not the purpose. It's really, as you say, there's, I think for many of us coming into this, we kind of thought it was one single, approach here. And what we find is that it's actually quite an amazing approach. I'm going to ask you a question that, really, when do you think, you 5 years now, Darren's been here, we're still kind of, you're just starting to negotiate patients. We see the long ramp up to sickle cell disease. We've seen the long ramp up to many of the others. Why does it take so long? And when do you think we might have something like the Fabries, in a position where you're actually doing an application?
Yeah, so they take a long time for lots of reasons. I mean, we've got to make therapies which are safe. And that takes a lot of preclinical testing to make sure that we're really satisfied that what we've got is potentially going to be safe in the clinic. And then we need to do the clinical trials in a responsible manner. So again, we are really checking safety. Safety is absolutely paramount, especially for Darren. I mean, you heard, he could go on with his ERT. Very different to Dr. Kaufman, who basically had the challenge of he'd run through all the oncology therapies that were available to him. So Darren had a choice. And so safety is obviously paramount to us. And then someone touched upon the topic of actually making the therapies available to everyone. So manufacturing is a very important component. And it takes a long while. These are pretty complicated technologies to manufacture. And we want to make sure every therapy works. And so it's that robustness takes time to create. And we spent a lot of time at AvroBio. And we started the company, we were really wanting to make sure that not only could we get great clinical trial data, but that we actually then could convert that into therapies available for everyone. And we're very passionate about that, making sure that we can make them at the scale that's appropriate, because thousands of patients have Fabry disease, as you know, thousands have Gaucher, thousands have Pompe. It's no good if we can only treat maybe tens of patients a year, because then everybody can't benefit. We've created something where people want and then they can't have, and that's just all wrong. So we really wanted to make sure that we could make these therapies available to everybody who needs them. So that's been one of the really big drivers behind the company. And unfortunately, that does take a lot of work. And that's why you see these therapies, they do take a number of years to get to the point to clinical trials. And then obviously the clinical trials typically take something like 3 to 5 years to go all the way through. And then the FDA takes about another 6 months to a year to actually review the final documentation. Are they happy that there is safety and efficacy and the manufacturing is in place? So it takes time, but we're going as fast as we possibly can. And it really is with the help of the whole community right behind us that enables us to do that.
Well, certainly when we look at the applications and the pipeline into the FDA and the EMA, I mean, the estimates are we're going to get something like 5 a year in the next decade. So it's a mind-boggling kind of a explosion, quite frankly. So this has been truly, there is a question here, and I don't know if you want to address it. I found it interesting. It says, from Marcos, are there other methods than a vector virus for getting the genetic information? So we did talk about zinc fingers before, but what about mRNA technology? Yes.
Yeah, so they're great questions. And I spent 30 years of my life trying to get genetic material into cells, because what we want is a durable effect. And yeah, the RNA technologies are really good. And we've seen them with COVID. I mean, without them, we wouldn't have the Pfizer and the Moderna vaccines, which have done such a super job. What they do basically is they don't integrate permanently. What they do is those mRNAs go into the cells, and then they're sort of instructions for making the proteins, which is what you want, but they're not long-term. If you want long-term, you actually have to have a piece of DNA that goes into the genome. So it's there forevermore. And so they're great ways of getting a burst of effect. So basically they go in, they produce the spike protein for COVID-19, and then your immune system sees it, creates an antibody and a T-cell response, and that's there hopefully for a good long period of time. What we want with our gene therapy is we want to actually integrate an extra copy of the gene with the right coding directly into the chromosomes, so that it's there for many, many years. And why do we think that's likely to be effective? Well, first of all, we know that HIV is there, unfortunately, for the life of the patient. Once they catch HIV, it's there because it's integrated in. So we know there's durable integrations possible. And then bone marrow transplants. Once a bone marrow transplant actually takes and engrafts, that's there for the life of the patient. So when you add those two together, this is likely to be a very long-term durable solution. And that's really how we see this. But mRNA is good for short bursts, works great for COVID, fantastic, but probably not appropriate for where you want sustained release of an enzyme over many years.
Great, thank you so much. And we certainly do have mRNA therapies, but as you say, they are continuous therapies. They're not the kind that you can, do once and be done with, in theory. Anyway.
Yeah, it's all about freedom for life. I mean, that's the whole vision behind AvroBio. Could we do once and done, and effectively impact patients so that they never had to go back to their ERT or need another therapy?
Well, we won't ask you the question that we have for the end of our workshop last time, and that is, what about sort of gene editing into germline cells? So that was a great conversation last time. We'll leave that in terms of the total transformation for another session. I just want to thank you so much, Chris, for your not only really cogent presentation, but your, answering the questions and bringing it all back home, based on what the patient said as well. So can't thank you enough for all of this. And really thanks to, this was not meant to be, this wasn't an infomercial for AvroBio. It wasn't an infomercial for lentivirus. And it was absolutely not meant to be a Canadian, but you can see that there's a Canadian strand to Brian, who was actually a physician here, to Darren, as you say, who got the first gene therapy up here, and certainly AvroBio, which actually had its beginning at the U of T. So this was an all-round Canadian triumph here.
Oh yeah, and we're very proud of our Canadian roots. Our CEO is from Canada, from Montreal, and we have our offices in Toronto. And the technology, as I said, is out of Canada in the first place. And that clinical trial we heard about was Canada. So yeah, we're very pro-Canada. And I'm delighted. So Rain, thank you very much for inviting me. I've really enjoyed participating. I should just say it's a team effort. There's 150 of us here at AvroBio working like crazy, but that doesn't include the huge numbers of patients who fire us up every day to get the job done. So thank you very much, and thank you to Cord.
Well, keep at it. We really, really need it. Thank you so much. Okay.
We're with you. Thank you.
Okay. So
thank you very much.
All right, so we're going to switch gears ever so slightly then and talk a little bit about global access. And we were trying to figure out how to do this. One of the reasons why we wanted to have this discussion is that we do know that getting access to these therapies is not straightforward. So what I thought I might do is actually switch over and and I'm going to give you a brief presentation. And rather than have us all go into, I think, separate breakouts the way we have in the past, we thought we would kind of keep everybody together and maybe have a bit of a discussion on what next steps might be. Certainly, next steps from the point of view of RDI, and next steps from the point of view of, what might be happening with regard to other strategies that we might have, but really, have been so very pleased in terms of the opportunity to have this series. And, hopefully we can think of some additional series that we might put together. We are very much, we'll reach out to you, get your feedback in terms of some other topics. And certainly the global access is one that we want to be able to continue having some conversations on. So I think I will share the screen myself if I can find this, and maybe take you through a little bit about, there's a lot of slides in here, but it's a fairly quick journey to talk about what we're trying to do, certainly from the point of view of RDI, Rare Diseases International, with regard to global access, but also I think, as you will see, some of the real challenges here. So what are the opportunities? This is a slide I use a lot, and those of you who were at our annual meeting may have seen it already. Really to talk about the fact is that, for just therapies in general in terms of rare diseases, is that we do know still only under 5% of rare diseases actually have a approved treatment. And given the 6,000-7,000 of them, it will be a long time to get to all of them, maybe. But as you can see, many of these therapies actually have multiple indications. This is what I think is already challenging. The figure came out, says under 10%, even in a well finance country like the US, an early adopter, only about 10% of people who should be getting a therapy getting it, and worldwide, of course, much less. We all know about the huge incentives that went into the Orphan Drug Act that really stimulated this huge amount of rare disease therapies that have come. And you've heard now Chris and others talk about the long ramp up in terms of gene therapies. I mean, these are, sort of beginnings of rare disease drugs, but, really we've seen now is that the focus around some of these durable long-term therapies. But the challenge, as we know, is having the drugs is not just that the drugs are there. The challenge is getting access. And this is just a snapshot and a little bit dated by now. But, what we do know is that, rare disease drug access is not evenly divided. There are, in fact, countries like France, which would be considered to be some of the very high access countries by all standards. Colombia, which has medium access, at least by global standards. They do have a special drug fund. There is no copay once you get onto a list in which your drugs are available. And then you've got, countries like China, which are moving very fast, but still would be considered very limited in the number of rare disease drugs you have access to. And part of it really is, the long challenge in terms of getting access to a therapy. It isn't just the cost, but it certainly is a whole lot more. And part of what this slide is meant to show is that it's actually quite somebody was talking about the bumpy road, is that there are many components to getting a drug for rare disease to the patient, which includes, obviously patients and everybody being aware that there's a therapy, having the appropriate kinds of healthcare systems and technologies to make sure that you can get people screened and properly identified, diagnosed, having the proper kinds of settings in which the therapy can be applied. And certainly something like a cell therapy or gene therapy requires a fairly sophisticated and a well-supported environment, both for the actual administration, but also for, as we say here, the aftercare. So, I mean, the challenges are multiple in terms of getting access to any of the rare disease therapies. But when you think about it in terms of something like a gene therapy, or some of the therapies that talk about something like a transformative therapy, which is another word, durable therapies, transformative therapies, these are these advanced therapeutic products we talk about them. In many cases, we're talking about therapies, as Dr. Mason's talking about, that you can have for either a single intervention or a short course of treatments that actually have significant benefits. So long-lasting therapies, which are going to, really impact both the progression of the disease, as Darren talked about, but also the quality of life. But the challenges there are, in fact, in the system, because in many cases, I mean, you heard Brian talking about the fact that he had cancer, and obviously cancer is a rapidly evolving disease, and gone through many courses of treatment before he ended up with CAR T therapy. But, we've had a lot of pushes, especially for, as these therapies become safer and as they become more, sort of more almost routine, should they not be earlier stage therapy before the patient has had a lot of damage. And even as Darren says, even with enzyme replacement therapy, your body actually gets continuous damage, both from the progression of the disease, which continues even with the replacement therapy. And the same with other, conditions like the hemophilia, like Gaucher's disease. The body continues to have a small progression of disease depending on how effective those therapies are. What about having that one-time therapy that could actually significantly slow, if not stop, the progression of the disease? I think that's the goal, but obviously these require then a whole lot more in terms of the health infrastructure to make them accessible, including a very accurate diagnosis, including these centers. And obviously, as we can see, certainly at this stage, certainly not trivial in terms of payment. As Brian says, one of the things they've done is set up a foundation so that they can do the education and even help support the financing of it, but also leveraging where there are insurance programs to make it happen. I mean, in Canada, as we say, we've got 2 gene therapies that have been approved, one of them for retinitis pigmentosus. Those of you who were with one of our earlier sessions heard us speak about that, had a great presentation around it. And also now with SMA, the gene therapy, Zolgensma, which, is in addition to 2 other therapies, but really is seen as that long-lasting therapy. I mean, in some countries, it has become available as a gene therapy. In Canada, we've got both therapies that are approved but not yet funded because of the huge challenges in terms of the financial impact upfront. And then, of course, the ongoing engagement there. So this is obviously not just the end of it. Notice I don't seem to have this in slide mode. Let me see if I can put it in slide mode. There we go. It's a little better. Okay, so I mean, certainly there are lots of systems, as we say, that are trying to figure out how do you actually make these successful to people, you know, and really the time is now, and we've got some systems that are starting 5 or 6 years ago to really think about how are we going to finance these, because it's one thing to say it's a full-time, long-term, durable therapy, and you can, in fact, be almost one and done. The challenge is that there's a huge burden up front. And most of it's certainly in the developed countries, have a cost benefits health technology assessment system that really challenges then that tremendous cost upfront. The systems are not really designed to be able to assess the impact of a long-term therapy, in part because, you heard me ask, what's your anticipation? Yes, we think that these are going to last for a long time. And as the clinical trials proceed over time, we're going to get more and more evidence that they will last for a long time, but we have no guarantees. We don't know, at what point might the gene therapy, the gene that's been, inserted, the gene that's been replaced, could it stop working? Could in fact something else happen? Could in fact the adverse effects become so overwhelming that it will not be workable? Certainly the early evidence suggests that it works, but when you're talking about a finance system that you're saying, okay, we want you to pay for something that's going to have a 10, 20, 30-year effect, but you're going to have to pay for it up front. Our systems just aren't set up to be able to do that. Systems are now beginning to think about how do we do this so that we don't end up with where we seem to be in the more advanced healthcare systems of trying to figure out how do we redesign our financing of these therapies in a way that's going to make them available. And really the word is going from how do we fund these therapies to how do we finance them? Can we pay for them over time? Can we pay for performance? And can we set it up so that it's almost like an amortization? As long as it works, we continue to make another payment. If it stops working and we need to switch the patient to something else, then we stop payment. Those are, different ways that we have never had before in terms of how we finance our therapies. The other challenge, though, is as we talk about global access, is where are they accessible? And I use this to say, part of the challenge, and as you hear, what Darren and what Brian and what Dr. Mason is saying is that we're still doing a lot of clinical trials. For a lot of patients, the access to it is actually getting it through a clinical trial. But not only for that patient, but also setting up those centers of expertise, getting the experience with the therapies, getting the healthcare systems and the financial systems to begin to think about how they're going to pay for it. So having clinical trials is really a huge, huge, importance in terms of getting these therapies more globally accessible. This is just a map that I use a lot to show that, clinical trials are not evenly distributed around the world. Huge bulk of them in North America, huge bulk of them in Western European, some in terms of the East, but a lot of that is really now China doing a lot. This is a map that I stole from a presentation, so don't tell on me because I didn't actually get permission to use this. I only got it recently. This actually looks at gene therapy trials, and you can see the same kind of an imbalance. If you happen to be in the US, quite frankly, if you happen to be in Western Europe, and this is because China is doing a huge number of gene therapy trials. The rest of the world, Canada, Australia, yes, to some degree, but many other countries in the world, it's not happening. So we're seeing the same inequity. And understandably, I don't think anybody's being deliberately biased about this, but this is the challenge if in fact this is the pathway for getting access in terms of not only patient experience, clinical experience, government experience. This is what we're talking about. These are the kind of barriers that already exist in low and middle income countries. That. Certainly there's a barrier in terms of the health care systems level. There's a barrier in terms of individual level, the lack of information about it, the lack of literacy about it, the inability to get a good, accurate diagnosis. But actually at the pharmaceutical level and at the international level, there is not necessarily that commitment to say, okay, let's think about how we're going to make them available to everybody. What is the strategy if these are in fact these life-saving, long-term, long-lasting therapies? And especially in those countries where there may in fact be, real challenges in having a good healthcare system, what if we could pivot to a therapy that was going to provide lasting benefits? I mean, this is the other challenge then. And there's some of the things that could be done. And this is not just for gene therapies, but for all therapies that we're talking about. The regulatory harmonization. If you don't have the capacity to actually do the approval in terms of risk benefits, could we not have a harmonized, almost a regional or global scheme for providing the approvals and to then be able to get the drugs into a country without necessarily that country having to redo its own regulatory assessment? How do we build up the health system capacity? And that's working across the world in that. Hopefully some of you have been taking part in the collaborative global networks that RDI has been working with the WHO in terms of really doing the initial stages of research on. We need to have regional rare disease centers, and we need to build up the capacity, especially in those countries where it does not exist, and certainly capacity for doing the diagnosis, but also high-level advanced therapeutic products should not be out of the scope of these kinds of centers. And then thinking about affordability. I mean, pricing is something that's a real challenge for rare disease drugs. It is obviously going to be that same challenge when we start to talk about these cell and gene therapies, which are a burden already in terms of developed countries, though not necessarily so if one thinks about the long-term benefits. Can we think about the pricing and financing models that are going to make it so that it is going to be accessible to other countries that are beyond the certain ones that we've got now, and then having more facilities for research and partnerships there. This is just a quick snapshot of saying there are lots of, we know, initiatives for trying to get the drugs to patients, and I think this is something that we can think about. Are there global initiatives? We can see that the global initiatives have huge holes in them. The COVAX initiatives right now are just, COVAX are not, are not getting the drugs to the, never mind, low-income countries, low and middle-income countries. There's not at all an equitable distribution there. Can you rely on, aid programs and charitable donations and sharing programs in order to assure that there's going to be equitable access? It's a huge question. And, certainly we can rely on a lot of these and all of these, but, is there another strategy that we need to think about when we think about some of these long-lasting, durable therapies that can have such a huge impact for patients, especially as we say again, where the healthcare systems themselves may not be in place. I won't belabor this. We did talk about this at our members meeting to talk about the fact that RDI has in fact taken on the task of a rare disease treatment access working group. We took that from ERDIC, which is the International Research Consortium on Rare Diseases, and took that on to really think about, and really start to put together strategies for ensuring that we're going to have access across the globe for these therapies, in looking at it in terms of three ways. What are the medicines that are going to be essential, starting with the essential medicines? What are the barriers, and what are the strategies for this? Again, I won't belabor it. I just want to introduce it to really say we are thinking about the ways in which we can address these barriers in a much more systematic kind of way. And again, I put it here for people that might be interested in participating with us as well as we're moving forward. Can can we work together collaboratively to think about this? And that was the end of that presentation. If I can figure out how to stop sharing, we would. All right, so that's sort of the end of the presentation, which gives us a nice half an hour, hopefully, to have a conversation with all of you. We have had, a lot of requests and a lot of comments and feedback on the series that we've been doing. And we've got a fairly small, concise group here. We thought we'd kind of leave it together all in one group and maybe open it up for discussions on your part. I was schooled by Angela to stop doing polls. People are not happy with polls. We don't need to have polls, that people know how to have conversations. So I'm going to open it up for conversation. What are your thoughts in terms of maybe first of all, some feedback in terms of the cell and gene therapies, especially since we knew, see so many more of those, and in the rare disease space, what do we need to do in terms of being able to think about a global strategy here? Some thoughts here, some concerns here. Two ways to do it. You can certainly put it in the chat, but I think as a group, maybe I would encourage people, if you don't mind, turn on your cameras so that we can see each other. And let's see if we can have a conversation here. I really appreciate the fact that people have been engaged, involved. They put so many questions in here. I think it's a great opportunity for us to maybe open it up into a conversation, and let's see if we can talk about it. So anybody who would like to, you can do one of two things. You can put it in the chat, but I have an even better option than that, is just raise your hand. Use the little, I think within your, somewhere in your, if you open the list of participants, you can raise your hand from the participants list. You can also, use the reactions box. There's a hand raising there. Or if you've got your camera on and somebody else is looking, just raise your hand physically and we'll try to recognize it. Dan.
Hello. Thanks a lot, first of all, for these nice workshops.
Can you introduce yourself first? I apologize.
Yes, of course. I'm Dan. I'm from Luxembourg. I'm with the National Alliance for Rare Diseases. And I've been following these workshops with great interest. So thanks a lot. I learned a lot, and I'm really happy to follow this. We've heard patients speak from the experience of gene therapy, and it sounds all great, even though it's probably a risky situation. But eventually, these are stories that turned out well. I don't want to spoil the fun, but do we have any testimonies from people where it didn't turn out so well? I have a bit the impression I get there is a cost-benefit risk that is strongly in favor of gene therapy, and it seems like, okay, let's go for it. But how can we grasp some of the downsides more clearly?
That's a huge question. I don't know if anybody else here is going to be able to address them. Those we do know, as some of you may know or recall, I do, because I was involved with hemophilia for so many years. And this was a good now 10, 15 years ago, where we actually had a child who died, a young person who died. And that stopped the clinical trials for a good number of years. Because it was, it wasn't a surprise, but obviously a death is something that needs to be taken quite seriously. So that has happened. As you've seen, or you may have seen, the recent trials for sickle cell disease from one of the companies was full stop, because the adverse effects were actually astonishing in terms of the unexpected numbers. So absolutely, I think there are risks. And I think that is the reason why, it was a little bit of a facetious question to Dr. Mason, why is it taking so long? It takes so long because we don't know what the long-term adverse effects are. We've just been getting feedback from some of the hemophilia studies, which were one of the first to go into gene therapies. It's a very straightforward gene replacement therapy. You boost up, the manufacture in the body of the clotting factor, and it's meant to have a long-term effect. We're hearing, and this is more anecdotal, but I think there is some evidence that, in fact, the effect is not as strong over time. So the amount of clotting factor that's produced by that inserted gene is not as long, doesn't remain. But it's still producing enough clotting factor that it works. So you don't need to be 100%, as a human being. So, I mean, I think that is the question, what are the adverse effects? And we are seeing, real challenges at times, as much as it doesn't work in some of them, muscular dystrophy ones, as well as we've seen some that says, okay, the adverse effects are problematic enough with people getting other kinds of conditions. And I mean, I'm not a scientist on it, so somebody else may have a better, understanding of it or some better examples. I wonder if anybody else has some other examples that they do know of in their disease area. Carlos, is that a hand up, or are you just kind of indicating that you're interested?
No, yes, yeah, I think that is, yeah, totally agree with you, Durhan. And that is the key question, what happened with the long term in terms of the efficacy and also the long-term adverse events. So far, we know that the key point in terms of the gene therapy is the immunological reactions. And as Durhan mentioned, in 1999, when he had the first case for to receive gene therapy, he died due to acute immunological reactions. And for that reason, it's important this is first part of the patient receives gene therapy, following very close in order to monitoring what happened with immunological reactions in terms of the cellular immunity, humoral immunity that is before to get infusion. For that reason, is not all of patients eligible to receive gene therapy, and then also the complement activation. Then in the long term, we need to differentiate what's happening with the integrative and non-integrative gene therapy. When talking about non-integrative is when you use a vector that goes to the nucleus, but the DNA is out of the chromosome. And this is the advantage of this is not integrated apparently, hypothetically is an hypothesis, maybe not have the risk of malignancies in the future. The integrative gene therapy that usually is ex vivo, for example, today with lentivirus, maybe have a potential to have long-term events in terms of the malignancies. For the reason now, the current environment, the regulatory point of view, we have in the non-integrative is 5 years follow-up, usually in the most common regulatory agencies, and the integrative or gene editing, or when using a specific vector that maybe is integrative in the future, you need to follow up for 15 years. Gene therapies started a long time ago, but the big stops due to the acute immunological reaction, then to develop leukemia, put on the table what happened with the long term. And for the reason now, if we're talking now, we need to wait until what happened. We need talking about durability also, what happened with the expression of different proteins in the current environment. Also, we need to thinking about what happened with the dose. Usually high dose, more immunological reactions, maybe you have an issues in the future. Is one program that is the XLMMM was stopped due to this problem with the high dose. But now is some uncertain things in terms of what happened, but definitely gene therapy, if everything is controlled, will be the future to treat this kind of monogenic diseases.
Great, thank you so much for that. Other thoughts or questions, or even in terms of, so it's very sobering, as you say, looking at the risk benefits still, it's early stages, but we do know there is certainly there are a lot of people that feel that this is in fact, the pathway in terms of long-term cures. Other thoughts in terms of either other disease areas, etc. Marcos.
Hello, I'm Marcus from Association of SYNGAP here in Germany, and the SYNGAP syndrome is not life-threatening syndrome, but is a syndrome which is very heavy for the affected persons. So they have a lot of problems with epilepsy, cognitive problems. They don't speak, and so on and so on. And but it's not a life-threatening disease. And if I hear what this gene therapies means for the patients, and I have to take the decision for my children because he cannot think for himself, and if I hear that he will be for a lot of time in a hospital for taking out the blood for this preparation, then for this preparation for this reinfusion of the blood, and I think yes, this approach with this long cure approach, maybe it's good for diseases which are life-threatening or life-shortening, but not for such syndromes like SYNGAP or Angelman, for example. Therefore, I ask about this mRNA
mRNA.
mRNA treatments, because the intervention is not so massive as the other methods, and I would prefer a more repetitive treatments, which is not so massive for the patients, than such a treatment like this one which we were presented today.
So again, that's sort of the risk benefits that you would see on it. We did have, some of the previous discussions around gene replacement therapies where in fact, you look at Zolgensma, right? It is an AAV vector. It is inserted, we were talking about, where and how, it can be administered, but quite frankly, it can be administered in a regular clinical setting. You don't discharge the patient in a few hours, but theoretically you almost could, right? Because there isn't anything that's taken out. In fact, it is put in. So it is, as you say, the difference between an ex vivo. So I take out the cells or I treat them outside, and then I put them back in, or do I insert the gene into the body and then the gene will be, functional within the body. And I think that's a big difference, at least from the point of view of those kinds of risks, as you say. And it may depend on the type of therapy. But again, I think as everybody says, you have to look very carefully and really have the conversations. What are the risk benefits? The challenge, I think, is, what about for people, anybody who says, are there other therapies that are available when you're talking about it for SYNGAP? Are there therapies currently available?
SYNGAP is a quite new syndrome. It was discovered in 2009, and currently we are looking for orphan drugs use, and we are looking also on similar syndromes which are more advanced with their approaches in change therapy. So there are talks with Angelman groups which are working on therapies based on ASO, this ASO method. But I think we are at the beginning of looking for new therapies, and we have a long way before us, and we are only currently 700 cases worldwide. So the number of patients is not very attractive for the pharma industry, and so we have a long way before us, and we can only achieve this by working together with similar groups or similar diseases, and now we're preparing some grants to participate in some grants, but we are at the beginning, so we are quite new in this scenario, and so therefore it's quite interesting to hear all this or get all this information, and to get involved in this world of gene therapies.
Yeah, I think that is getting to be certainly a great approach, right? Where you look at very similar diseases, and can we really think about how we're going to create treatments that would be across a range of diseases? If we go after 6,000 or 7,000 diseases, it would take us forever, but we are making great advances there. Can I just ask one other question? Is it in fact a monogenetic disease? Do you know?
SYNGAP is a monogenetic disease. It's on the sixth chromosome on a certain place, and it's a monogenetic disease.
Okay, but it certainly can be quite pervasive, as it says here. I think you've got a response there as well. Does anybody here actually have experience with either trying to get access to gene therapy, or know of patients that have had gene therapy, or how gene therapy is being available in your country, that can share, what are some of the challenges in terms of getting access to a gene therapy that may indeed be available?
I'm Luc from Belgium, Duchenne Parent Project Belgium. I think the big
Thanks, Luke.
hello. I think the big challenge will be HTA, because very few or no states will be able to pay millions of euro, as you mentioned, in upfront, without knowing what happens with the cure. Does it work for 10, 15, 30 years in the case of Duchenne, for instance? I think we have to have a global view to how to suggest a dynamic reimbursement over the years. I think it's essential, otherwise states won't be able to pay, and patients won't be able to be cured.
Yeah, I mean, and let me ask you in terms of Duchenne, because it's it's one of those diseases where, good gosh, we've known the gene forever. We know how the gene works. And yet, it's been such a challenge, right, to getting a therapy that really, really does work. And I know a lot of hope is being put into gene therapy. I mean, where are you in the thinking of that, because I just saw, somebody sent me a clinical trial results from a very, very, promising clinical trials, phase 2 clinical trials being done, but again, a more traditional therapy. Where's the community thinking in terms of their hopes? Is it in terms of maybe we will get something as a biologic therapy is coming through? Or are we really thinking that it's going to take a gene therapy for us to get to something?
At the moment, for Duchenne, it's the only hope, because other therapies are just light remedies to replace corticosteroids with less side effects than steroids. But it's not a cure. The only hope for a cure is gene therapy. So it's really in some cases mRNA can be useful, but only for a certain percentage of the population. For a largest percentage of the population, it's gene therapy or nothing.
Yeah, I
So
think
it's
there was a lot of
essential.
hope around some of the chromosome skipping kinds of therapies that were coming in, and the nonsense.
Works for 15 or 20% of the population.
Yeah. So, but as you say, the challenge really will be getting financing models in place. I mean, the good news is that we are seeing Europe, and certainly I think your consortium, they're also trying to come up with models that are going to be thinking about, as these therapies come in, what's the financing model in order to make it happen?
And the other concern we have is, this European regulation about GMOs that is putting a big brake on trials, because as it is not really well defined, this it's a frame, as very often in Europe. A lot of states can interpret this frame as they want. And in Belgium, for instance, we see that it's a big break on therapies, because everybody or anybody can ask questions about GMOs, but it's not the intention of spreading GMOs in the nature. It's just giving a therapy to one person. It won't even repeat probably to the descendants. So I think it's also a problem in Europe.
Yeah, the misinformation, the disinformation, that has nothing at all to do with the kind of research we're trying to do. The other thing I found startling around Europe is that every European country has its own rules and regulations about clinical trials with gene therapies, and about the approval I mean, so, it's making it isn't as if, it's not like the regulatory approval. You go in, you get an EMA approval, and off you go. In this case, you really are trying to deal with, various, and even some regions within the countries, which are putting big barriers in terms of the clinical trials. So I think that's a huge issue. Sally, let me bring you in the conversation here.
Yes, I'm Sally from the UK, but I live in Germany. Also part of the Duchenne community. I'm on the board of the World Duchenne Organization. I just wanted to say we in the Duchenne community also need to be very careful about talking about a cure, even concerning gene therapy. What we know or hope is that it can stabilize or slow the progression. We don't even know that yet, right? But what we are pretty sure that it won't do is repair any of the muscle that has already been damaged. So we really need to be very, very careful. And the other challenge is, I think in particular, is the duration, how long it's going to last. We've been seeing in the current phase 3 trials, well, the one phase 3 trial that's ongoing, that a huge number of potential trial participants can't take part because they have pre-existing antibodies. So that is another huge issue that means for the whole Duchenne community, it's going to take an incredibly long time. And as for the payment, I totally agree with Luke. It's complicated and difficult, and currently I don't see how it's going to work, but it's going to, there are going to need to be changes in how these therapies are financed, should they eventually come to the market. So
well, kudos to the World Duchenne community, though. You folks have actually been the leaders in the patient world for taking on, bringing together globally the Duchenne world. We know that there are so many, Duchenne groups, and you've been doing an amazing job of having people kind of come together. Also, the education that you're doing within the Duchenne community. When we hear you speak with the kind of knowledge that you have and the kind of concerns that you bring into it, I think it really will make for much better informed kinds of decision making. I think there's the notion among policymakers that, oh yeah, everybody's going to want to jump into this. Now we're going to be stuck, we're trying to figure out how we're going to finance all this once. And what we realize is not that way. People are very deliberate about it, even as you say, and as Luke was saying, where there aren't any other effective treatments at the moment, this might be the best, but, we cannot just rush into it. But I think the other concern you had said is something that's very scary, and that is so many people do have antibodies already, and what's the option then if that's going to be the case, because you can't do the clinical trials. We don't have enough patients to do the clinical trials. And, and I think you guys coming together again to say, what, we have to work as a community. We cannot have each and every company that's coming in, siphoning off a patient here, siphoning off a patient there. And all of a sudden we find ourselves, with a whole bunch of trials that will not necessarily lead to that meaningful impact. So good on you guys for the work that you're doing in terms of bringing communities together and showing how you can, do that collectively. My hat's off to everything that's being done in your community. And I think a lot of it with volunteers that are coming into it. It's not as if you've got a huge paid staff that's running these in that same way. So it really is a great example. And right around the world, you're bringing them in. So again, huge kudos to what you folks are doing.
Thank you for that. We do try to do our best, but we are also still learning, and particularly gene therapy. There still is a lot to learn, and we just have to be careful about things that are just not going to come true and just lead to total disappointment, particularly where access is concerned. It's all very well to have a fantastic gene therapy that nobody can pay for, so nobody can get, right? So,
well,
we have a lot of work to do.
But it also brings us back to one of the fundamental things we need to do is to be able to do early diagnosis. We've got to get, the newborn screening. We've got to get the early genetic screening, because the big hope, as you say, is can we get that child before they've developed, disabilities, to be able to say, okay, we can treat them now, because that's the best hope that we've got. We cannot repair, but we can, with each, group there. And that's, where we've seen SMA do so much, right? They've been able to now. We have a newborn screening. Now we got to get people to pay for it. And then we got to get them to act on it. So it's all bits and pieces. We do come to the end of a time, and I know that people will need to leave. I really want to thank everybody who's been on this session and the ones that have been here before. This has been a great opportunity to really begin. For us, this was an experiment. We had no idea. We'd never done gene therapy and cell therapy workshops before. We didn't know if it was too early or whether or not we were going to be, kind of fanning, faint hopes that we're not necessarily there yet. But we do know, and listening to especially what Sally and Luke are saying, right now, and then Dan, the timing is now, and the timing isn't good in order for us to be able to push this forward. So we thank you so much for having participated. We're going to reach out to you to do some surveying, and we're really hoping to do maybe another series that we'll start look at things like genetic testing, look at genome sequencing. What about getting into the diagnosis end? Because we have to start talking about that as well if we want to get to some meaningful gene therapy. So thank you so very much for participating. Huge thanks again to Pfizer for the sponsor. Thanks so much to AgroBio for staying in with us on this. This was wonderful, and really a huge contribution in helping round out our understanding. I would appreciate it, anybody, if you want to provide feedback on your own, send us an email, send us a note, tell us what else you want, and I'm sure Kalani is going to be reaching out to you for a follow-up. So thank you so very much for having made this such a very, very successful series of workshops for us. Really, really all the best. And we will see you again, in our next series of workshops, discussions, forums, etc. So do continue to participate. And if you're, need any more information, do reach out to us. Thank you very much.
Thank you. Bye-bye.
Thank you, Kalani, for all that you've been doing in terms of getting this going. Thank you, Angela, for making this happen. Appreciate it. Thanks, Lori.
Bye, everyone. Thank you. Bye-bye.
Bye-bye.